Happy Friday, 4 weeks down. So I have the pleasure to introduce Raúl González-Pech as our presenter today. Obviously, his work is very much interest to me, given that I spent my life studying the Intracellular Eukaryotes. And I think Raúl and I decided that he would do some of the details of his life and introduce himself. So I will let him do that. But before I give the floor to Raúl, I just want to stress that the endeavor to do comparative genomics on these symbionts is not easy. Remember, these are not bacteria. So wipe that idea out of your mind. These are single-cell eukaryotes with very, very massive genomes. They're approaching the size of the human genome. They're an order of magnitude bigger than animals. So you have to ask, who's the master of this house? So with that, Raúl, looking forward to your talk. He's going to talk about past, present, and future.
Yeah. Thank you, guys. I'm very happy to be here. Can you hear me well in the back? Yes? Okay, great. Awesome. So I'm very happy to have the honor to speak to you about my research. Today, I'm going to tell you a little bit about one of my major scientific interests. But before that, I would like to tell you a little bit about myself. So I'm Mexican. I was born and raised in Mexico. And this is not working. There. Nope. Not working. Yeah, it was working before.
Okay, continue talking, I'll figure it out.
Yes. And today is actually a very special day for us Mexicans because we're celebrating the Independence Day. And that's also why I'm wearing this nice traditional shirt. This is actually the main holiday in Mexico, not Cinco de Mayo, but today. So it's a great coincidence that this is happening. Yes. That's fine. No.
I don't know why you can't share your screen. What happened? Should be good. Try clicking.
No.
No.
And this
All
is
right, I got it for you.
okay.
Right, I got it. I will click for you.
Okay. Thank you. Yeah. So I can get rid of this. Well, a little bit more about myself. So I was born in Mexico. Then I did my undergrad in the north of the country. Yes. Yes. And that's where I started working on coral genetics. I did my thesis in intracolonial genetic variation in a species of corals. Then I moved to the master's to Germany, where I started doing some not only work on evolution, but also work in bioinformatics and genomics. And after that, I went to the Ph.D. In Australia, where I started working on the genomics of these coral symbionts from an evolutionary perspective. After that, I came to the US, where I continued to work on genomics of these amazing symbionts, but also we incorporated some work on epigenomics. And now I'm here at Penn State, and I've been here for about 6 months, which doesn't seem too long, but actually there are so many things happening here, and I'm very excited to be here doing some amazing research. But let's go back to the topic of this talk. So today I'm going to tell you about how coral symbionts, and I hope I can convince you, are good models to study impact of symbiosis, of intracellular symbiosis on genome evolution of microbial eukaryotes. And I know this, is it working? Awesome, great.
Here, you're clicking, it's right here.
Yeah, but I'm trying to go back and.
It is.
Yeah.
Okay. Work on my hand.
It's my hand. There's something wrong with my hand.
Okay, it only works for one. Okay.
Only works.
Yeah. Go ahead.
All right. Right. So if we go back a couple of slides. Yeah. So I know it's a very long title. So throughout this talk, I'm going to try to break it into smaller bits. So first, let's talk about symbiosis. And we all know that symbiosis stems from the Greek sym, which is pretty much together, and then biosis, which is just living. So symbiosis means symbiosis is pretty much any relationship, any type of relationship between organisms, very close, very close-knit relationships. And really, symbioses are everywhere across all domains of life. And we have even had some talks about symbiosis in this seminar. The very first talk by Seth, he was telling us about the importance of the Wolbachia symbiosis with many insects and other arthropods. But symbioses are also very important for humans from many different aspects. For instance, if we think about health, like the human gut microbiota, or if we think of agriculture and food safety in terms, for instance, of rhizobia that are important for the growth of plants and some of the products that we consume, and also from an environmental perspective. And we're gonna be touching a bit more on this later on. Yeah, thank you. And now let's talk about how symbiosis can impact genome evolution. So we know that organismal interactions can actually drive evolution, and perhaps the most evident example is coevolution. And here I'm showing a very textbook example. This is Darwin's orchid, in which we can see, or we can appreciate that this interdependence between these two organisms has led to their evolution into very extreme phenotypes, morphological phenotypes, right? And however, coevolution can also have impacts in the genetics of these organisms. And this becomes very clear when we look, for example, at the evolutionary arms race between hosts and parasites. This is also something that is known as the Red Queen hypothesis, because they have to keep on moving to stay in the same place, right? But symbiosis can also affect evolution, and more specifically genome evolution. But this type of effects or this type of impacts are not as direct there, or they're not really driven by selection and adaptation. And here I'm going to show an example based on intracellular bacteria. So here we have a representation of the genome of a free-living bacteria that is close relative to this intracellular bacteria. And we can say that these genomes are quite regular or quite normal. Nothing very interesting happening. Okay. Yeah. No, there's something with my hands.
Yeah, your hands.
Yeah, my hands. Yeah. I'm clicking that one. Yeah. I think it might be just the signal then.
Try it.
So here it works.
Yeah.
But here? Yeah, I think it's just,
Okay.
I have to go like this, I think.
All right, I got it.
Yeah.
Okay.
Or,
Just
yeah.
look at me, point at me.
Okay, thanks. Yeah. All right. So we were saying that the free-living relatives to these intracellular bacteria have quite regular genomes. But when the early transition to the intracellular space, the early confinement to that intracellular space happens, then a series of changes start to occur. And this phase is really characterized by extensive structural rearrangements and enhanced activity of mobile elements, many duplications of genes and many other types of elements, accumulation of pseudogenes, and so on. And as, can you go back? Yeah, thanks. And as time goes by and these bacteria remain confined to the intracellular space, many of these replicated elements become redundant within the same genome and also with the genome of the host. So there is a relaxation of selective pressure that causes many of these replicated elements to degenerate very quickly. And this causes mutational biases and deletion biases. So many of these elements get wiped out of the genome over time, and the genomes start becoming smaller and smaller until very extreme cases that we can encounter in genomes of obligate symbionts and parasites. And perhaps some of the most extreme examples are the genomes of mitochondria and chloroplasts. Next, please, yeah. But now this is an example that we know. This has been clearly described in bacteria, but what about intracellular eukaryotes? Does this also happen in these organisms? So there is some evidence that suggests that this is the case. And here I'm going to show a couple of examples. This first one is presented by nucleomorphs. And these nucleomorphs are the degenerated nuclei of eukaryotic cells that are secondary plastids. So it's not only a case of primary endosymbiosis, but a secondary endosymbiosis in which the eukaryotic cell has been engulfed, right? And these nuclei are very reduced, very vestigial, and it's observed in a couple of algal lineages. So indeed, genome reduction can happen in eukaryotic intracellular eukaryotes. Next one, please. And then another example is posed by Microsporidia. And this is a very interesting group because they have very, very derived genomes. They're intracellular parasites. And it was very difficult to position them into the phylogenetic tree of life because of how different these genomes, how diverse these genomes are. And now we thought, and initially we thought that they were protists, but now we know they're actually more closely related to fungi. So really, intracellularity can cause this divergence in this type of genomes too. And now, oh, sorry, yeah. Yeah, I can try, yes. Yeah, otherwise we can try to kind of tilt it.
Yeah.
And maybe use the clicker. There we go. Yeah, yeah. It's just it was blocking, I think, the podium. Yeah. Okay. Good. So that's working. Okay. So now I'm gonna try to convince you that coral symbionts are a good model to understand how these changes occur. But first, let's talk a little bit about these coral symbionts. So these symbionts are dinoflagellates, and they belong to the family Symbiodiniaceae, which means that they're microbial photosynthetic eukaryotes. They are microalgae. And they are not only symbionts of corals, but also of many other marine organisms, and there are even some exclusively free-living taxa. They're commonly referred to as zooxanthella, but this is a term we try to avoid because there are also many other marine eukaryotes or microbial eukaryotes that live inside some other animal host. So we prefer to avoid that term. And they were initially described as a single species, but now they're recognized as a very diverse group. And it's funny because whenever I tell people that I work with coral symbionts, they immediately picture something like this. Like this. But I'm a genomics person, so in reality, I spend most of my days trying to make my code work and try to make sense of huge amounts of sequencing data from a biological and evolutionary perspective. But I do enjoy going to the water every now and then. But let's go back to talking about Symbiodiniaceae. The reason why they were described as a single species is because they are pretty much look the same. They don't exhibit such a great morphological diversity. So if you put them under the microscope from any sample of Symbiotic from reef around the world and put them under the microscope, you'll probably see something like this. But they do exhibit some subtle differences, for instance, in their cell sizes. And here we're just showing their lengths compared to their width. And we can see that there are some differences for different clades or different genera. There's also some difference, for instance, in the way that plates that constitute the cell walls are arranged on the cell surface. However, the diversity becomes more evident when we look at their ecology and genetics. Here we have a diagram that represents different types of symbionts and non-symbionts. So for instance, number 1 is showing us the typical very dominant symbiont of, say, a coral that occurs at higher abundances. But there are also some that occur at lower abundances in the background. And for these ones, we don't really know what the nature of the symbiosis is, if they really engage into real symbiosis or what they're doing in there. There are also some cases of extracellular symbionts in some hosts. And then there are even some taxa that are exclusively free-living, as we mentioned before. In terms of their genetics, thanks to the use of molecular markers, we know now that they're very, very diverse. And here I'm just showing a series of episodes of adaptive radiations of one genus. This is just genus Cladocopium in the Indo-Pacific and the Atlantic Oceans. And we can see that there's only 2 ancestral types that cause the huge diversity that we see in these 2 oceans. And some of these adaptive radiations even gave rise to groups of symbionts that became specialists to certain coral taxa. When we talk about diversity of the symbiosis, first we have to consider that all the symbiosis between coral and symbionts and also between the symbionts and other organisms is of a nutritional nature, right? So there is a great metabolic complementarity between the 2 parties. But even though there is a lot of variation in the types of symbiosis that we find in this family, and we saw it in the previous slides, but there is also variations, for instance, on how specific the symbionts are. Some of them are generalists, some of them are very specialized on the transmission mode from host to host. Some of them are transmitted vertically from the parents to the offspring, and some of them are acquired horizontally from the environment. And there are even some that have mixed transmission modes. But for the purpose of this talk, we're just going to consider the spectrum of symbiotic associations from this perspective. And these are three broadly defined categories and probably very oversimplified from what occurs in nature. But we're going to be considering those that are exclusively free-living, and then a wide of facultative symbionts. And these symbionts are the ones that can survive both inside the coral host or any other host, and also in the environment. And we actually have many of these in culture, even here at Penn State in Todd's lab. And it's funny because I was talking with colleagues, and some of them, when they think about coral symbiosis, they don't realize the side of the symbiont perspective, right? Because for the coral, the symbiont is obligate, so it's not facultative. They really need the symbionts to survive. But for symbionts, it's not always the case. But then we also have some symbionts that occur rarely in the environment and that are mostly transmitted vertically from parents to offspring, and that we have been struggling to culture. So we can think of this as obligate symbionts. So that, oh, sorry, I forgot to mention that these three categories occur, they're repeated across the phylogenetic tree in different positions of this family, which is great because we kind of have natural replicates of these three different categories along the phylogenetic tree. So it's like a natural experiment where we can do some comparisons, right? And that's what makes them a good model to study evolution of their genomes from a symbiosis perspective. But also, it is very important to study these organisms given the current coral crisis that we're facing, in which we have mass coral bleaching events. And coral bleaching is nothing else than the breakdown of the relationship between corals and this microalgae. So hopefully now I have convinced you that Symbiodiniaceae are good models to study genome evolution of intracellular eukaryotes. However, this is not entirely true. And that's because what Todd was saying just before, dinoflagellate genomes are actually very derived, so very challenging to work with. And here I'm just going to list some of the features that make them very special. So Todd mentioned how big they can be. And here we have an example of Procentrum micans that has a genome size of about 275 billions of base pairs, right? And then when we compare it to the human size of the human genome size, well, that's a big difference, right? Their chromosomes are also very peculiar. They have this fibrillar structure, and they have this liquid crystal appearance, and they're also permanently condensed. And this compaction is not caused by histones, but by histone-like proteins from bacterial and viral origin. Their genomes are also very repetitive. This clicker. The genomes are also very repetitive, and what I'm showing here in this micrograph is probably a very extreme case. So this is a fluorescent in situ hybridization, where the probe consists exclusively of simple repeats, ATs. And it binds, you can see that it binds an entire chromosome. Here is a replicated state of the genome, but it's an entire chromosome of simple repeats. What is that genome, that chromosome doing there? We don't know. Oh, and then their genes are also quite interesting. They're commonly arranged in these clusters, facing all in the same direction and sitting on the same strand. They also have unconventional intranet exon boundaries. So for most organisms at the 5' end of the exons, we have a G and a T. But for Symbiotic and other dinoflagellates and all dinoflagellates, we have G and then followed by any of the other nucleotides at about the same probability. And then at the 3' end, for most organisms, we have an A followed by a G. And in dinos, we have an extra G after. There is also a hypermethylated status of these genomes, especially when we talk about CpG's motifs. And then we even have a 5th nucleotide that is hydroxymethyluracil that replaces many of the thymines that we have in the genome. But the main message of this slide that I want you to keep in mind is that dinoflagellate genomes are very, very weird, right? And when we work with these genomes, we have to take some of these features into account. And this is an example of that. This is a pipeline that we established to do gene prediction from these genomes. And we had to modify these programs to account for those unconventional intranexon boundaries, right? And a question that I had during my PhD dissertation was whether these intracellular symbioses could be driving some of the genetic diversity that we observe in the family. But however, we know now from this talk that really the genetic changes that symbiosis can cause are at the genomic level. So here we can cross out genetic and look at the genomics. But a few years back, we didn't know much about the genomic diversity of this family. We didn't really know whether the genetic diversity that we see based on a few gene markers really scale up to the whole genomes. So the first insight that we got from this was from the analysis of transcriptomic datasets, because a few years ago, it was quite difficult to access and to sequence some of these genomes. So what I decided to do was collecting a bunch of data sets that were generated from previous studies and looking at gene families that were shared and unique to the different lineages or to the different genera in the family. And that's what I'm showing here. So this is an offset plot that is showing all possible combinations here at this bottom panel of all these lineages. And here, just on the top, we see a bar plot showing the number of gene families corresponding to those combinations. So for example, the first column is showing just the gene families that are exclusive to Cladocopium. The second column, the gene families that are shared by Cladocopium and Microbiome, and so on. And something that immediately pops out is that lineage-specific gene families or genus-specific gene families are very abundant, whereas gene families that are shared by all genera in the family are way fewer. So this is already telling us that there are extensive differences in gene families among these lineages, but also that there is extensive genomic divergence within the family. And this was later corroborated with the generation of whole genome sequence data from my dissertation. And here I'm just showing a phylogenetic tree, a cladogram of these genomes that we had available and that we generated. And at each node, we see the number of gene families that are shared by members descending from that node. And we can see that there is a lot of variation still. And even when we look at gene families that are exclusive to each of these genomes, we can find some differences. Even among members from a single species, we can find gene families that are specific to that one genome. So again, this is striking. But now that we had whole genome sequence data, we could look at diversity from different perspectives. And perhaps the most intuitive way to do it is looking at the DNA sequence itself. And this I did by doing whole genome sequence alignment, done pairwise between two pairs of genomes at a time. And that's what I'm showing here. So on the X and the Y axis, we have the same genomes treated as queries and as reference. And each of these blue squares is telling us about how good the whole genome alignment was for that pair of genomes. And the color is representing the percentage similarity of that alignment. So the darker, the more similar the two genomes. And the size of the square is representing the fraction of the query that aligned to the reference. And we can see from this diagonal, those are very good alignments, pretty much 100% similarity and 100% of the coverage of the query. And that's because this corresponds to each genome aligned against itself, right? But what I want you to see is that there is a lot of diversity again. There is a lot of variation among these genomes. And even if we focus on these few samples, these are all from the same genus, right? And even here, there is a lot of variation. And if we pay attention, we'll notice that some of these alignments are comparable to alignments between distinct genera in the family. Another way to assess genome diversity is looking at the composition or the structure of these genomes. And here I'm going to show an example of blocks of genes in the same order that are conserved among these genomes, and again, done pairwise. And this is a chord plot in which each chord, or the width of each chord, is representing the number of gene blocks shared between, again, pairs of genomes, two genomes at a time. And this is actually the largest combination, the largest comparison, with the highest number of gene blocks being shared between these two genomes, when actually these two species have multiple strains represented in this analysis, right? This is the second largest comparison, again, between two different species. And then this is all the possible comparisons. And it's interesting that even we see some cases in which the band or the string is so thin that represent only very few gene blocks being shared between pairs of genomes. And these are all members from the same family, except this outgroup species, right? So again, the genome divergence, the genome diversity in the family is tremendous. Now, when we talk about diversification of Symbiodiniaceae, several aspects have been considered. One of them is, for instance, ancient events. And here we have the most accepted phylogeny of the group at the time, with estimates of divergence times based on the molecular clock technique. And here at the bottom, we see a timeline that shows the diversity of scleractinian corals, hard corals over time. And something interesting is that the origin of the family matches an episode of adaptive radiation of these corals. We suggest that these two groups have influenced each other's diversification, right? Another aspect is ecological specialization, so adaptation to a particular niche. And here what I'm showing is just how some lineages of Symbiodiniaceae have evolved to be specialized symbionts or symbionts specific to certain host taxa. Finally, another driver of diversification that has been proposed is sexual recombination. Even though we didn't really have evidence that this was happening in Symbiotic, it was only hypothesized to be so. But now, last year, this study actually showed some direct evidence of sex happening in the family. But what I'm proposing is that symbiosis can also play a role in driving this diversity. And again, when we think of the three categories that we mentioned before, what I did next was drawing a series of expectations of what we might encounter in these three broadly defined categories. And this is what I proposed. So for free living species, again, we would have pretty regular genomes. This is again based on what we know from other systems, so mostly bacteria. Facultative symbionts would instead resemble that early transition to intracellularity, where there is with this very dynamic phase, with this abundance of structural rearrangements and mobile elements and pseudogenes and so on. And obligate symbionts would instead resemble that reductive phase, right? That genome reduction with way fewer of these genomic elements and smaller genome sizes. And this is great. Now we have a series of expectations. Now we can start doing some comparisons between symbiotic and free living species. However, we have an issue that most of the genome data sets that were generated at the time were based on short-read sequence data. And why is this an issue? I'm going to explain this very briefly. So when we do genome assembly, this is what we usually do. We have a genome of interest. We have our sample or our organism of interest. We extract the DNA. We shear the DNA. And we send it to sequence. And then this is what we get back. Oops. A bunch of reads, right? A bunch of billions to millions to billions of reads, sequence reads that we have to put back into the puzzle together. And the first thing we get, and we do this, of course, computationally, but the first thing that we get are these very contiguous sequences, right? Known as context. But then oftentimes we have longer range information from these sequence reads that we can use to tell which context would belong with which one and in which order. And then we put them together and we insert arbitrary gaps between them. And these sequences, these longer sequences, not necessarily as contiguous, we know them as scaffolds. But then I mentioned that short reads are an issue. And here I'm going to compare, I'm going to show why compare to long reads. So imagine we have this repetitive element. So this is a repeat in tandem. You can think of it as a gene repeated in tandem. And when we get short read sequencing, when we do short read sequencing, this is what we get. So the reads from the end, those are not very problematic because we can tell them apart. This region is probably different from this one. So we know that they belong there. But all these regions, all these reads from in between, those are very similar, if not identical. So then when we try to reconstruct the sequence, the original sequence, this is what we end up with. And we end up with this collapsed repeats, right? With high read coverage. However, if we have reads that are, excuse me, that are long enough to span that entire repetitive region, then we don't really have to be concerned about this. And that's the beauty, and that's the importance of long reads. And I mentioned that most genomes of Symbiodiniaceae are short reads, even nowadays. And now what I'm gonna do is show you, excuse me, a comparison with some genomes of high quality from the Vertebrate Genome Project. So what I'm gonna be showing you is their distribution along these two axes. And here I'm showing you scaffold M50. And M50 is like a median of the sequence length, just adjusted a little bit, considering more giving more weight to the longer sequences. And I'm showing that for scaffolds and contigs. But remember, contigs are really what tell us contiguity. And this is where the vertebrate genome project genomes are. So far up and right. And the genomes of Symbiodiniaceae that are based on short reads are on this end of the spectrum, right? That's quite a big difference. These are logarithmic scales. But when we incorporate long read sequence data into these assemblies, and these are hybrid assemblies mixing both short and long reads, we see an improvement of the quality in terms both of scaffold and contig M50. But there have been even some efforts to generate chromosome level assemblies of Symbiodiniaceae, but they didn't really put any effort into generating long read sequence data. So their scaffold M50 improved a lot, but really the contiguity that is given by the contigs remains about the same, right? And actually, these two genomes are the ones that we use for the next comparisons that I'm going to be showing. At the time we were doing this, we didn't have enough funds to generate long read sequence data for all the genomes we wanted to compare, so we only selected two. And we selected the genome of a symbiont, and this is Symbiodinium tridagnidorum. It's typically a symbiont of clams in the Indo-Pacific. And this particular isolate was actually recovered from a coral from the Great Barrier Reef. And the free-living species is Symbiodinium natans. This is commonly found all over the world in environmental samples. And it has failed to infect hosts experimentally. So we're pretty confident that it's a free-living species. And this one isolate was recovered from seawater from Hawaii. And remember, these are hybrid assemblies that allow us to do a more in-depth comparison, right? And this is what we found. This is a summary of what we found. Here I'm showing on the x-axis different types of genomic elements. And on the y-axis, I'm just showing the representation of these genomic elements in the two genomes. So this is just a ratio of the length, the total length that these genomic elements occupy in the genome of the symbiont relative to the one of the free-living species. So if we have bars that are sticking out to the positive side in pink, those are genomic elements that are more represented in the genome of the symbiont. And this one that is sticking down to the negative side is in blue, is representing those elements that are more element in the genome of the free-living species. And then we also have this other bar, and this is the difference in genome sizes between the two species. So it turned out that the symbiotic genome was 1.74 times larger than the free-living species. And so that's the actual ratio. That number in each bar is the actual ratio. But what I'm showing here is a log2 transformation of the ratio. And we immediately see that some of the genomic elements that contribute the most to these different in genome sizes are like different types of duplicated genes and different type of repetitive elements that are associated with mobile elements. And we can look into some of these with a bit more of detail. So because we found a lot of way more duplicated genes, different types of duplicated genes in the genome of the symbiont compared to the free-living one, we expected to find more prevalent gene family expansion in this genome, in the symbiotic genome. And this is what I'm trying to show here. So here on the x-axis, we just have a ratio of the gene family sizes between the two genomes. And the y-axis is just telling us significance from the Fisher exact test that we did in the comparison. And again, if the circles are in pink on the right side, those are more gene families that are larger in the symbiotic species. If they're in blue on the left side, those are gene families that are larger in the genome of the free-living species. But only those that are filled are the ones that were significant. And if we see most of these looks like pretty symmetrical, so there is really no differences in gene family sizes between the two species. But the gene families that were larger in the symbiotic genomes had functions related to biosynthesis of glycoproteins, protein transport, and ankyrin domains. And all these functions are relevant for symbiosis, which makes sense. But why could this be happening? Why we don't really see any differences in terms of gene family sizes between the two species? So the answer could possibly lie on the fate of duplicated genes. So duplicated genes can keep their function. So if the function remains, then their sequence remains similar, and then this would contribute to gene family expansion, right? However, if the function changes, then the sequence starts becoming dissimilar. And depending how dissimilar these sequences are, this can cause that our algorithm fail to cluster sequences that actually belong together as gene families, right? And evidence of this we found in single copy genes of the two genomes. And we can see that there are way more single copy genes in the genome of the symbiont compared to the free living one, right? Another alternative, oh, I forgot to mention that actually we found evidence that these two set of genes share some similar proteins domains, which could be like vestigial functions, right, from the original sequence. Another alternative for the fate of duplicated genes is that the function is completely lost. And in this case, the sequences degenerate quite quickly. Of course, they wouldn't be classified as members of any gene families. But what we did to look for this was doing a pseudogene screening in the two genomes. And again, we found more pseudogenes in the genome of the symbiont compared to the free living one. Oops. Another thing that we saw more represented in the genome of the symbiotic species is where repetitive elements that were associated with mobile elements. And these repeats are scattered throughout the genome. And to study this type of repeats, it is very common to do this kind of plot. This is an interspersed repeat landscape in which we show some measurement of divergence on the x-axis. In this case, it's chimeral substitution level adjusted for CPGs. And on the y-axis, just a fraction that each repeat occupies in the genome. And here we show in different colors, different types of mobile elements, different types of repeats associated with these mobile elements, and some that couldn't be classified. And these plots can be very informative. So for instance, we see this peak at a relatively high divergence level. And this tells us that this is an ancestral burst of this type of genomic of mobile elements, right? That is shared by members of the genus. And this is actually very prevalent in the entire genus. But more interestingly, we find this peak of conserved long terminal repeats. Again, it's a repeat associated with some type of mobile elements. And they are at low divergence. And what this means is that probably these elements are still functional in the genome. And actually, there is some literature that suggests that these type of elements get activated under thermal stress in Symbiodiniaceae. And again, we can see that this type of repeat is more represented in the genome of the symbiotic species, right? So these conserved LTRs are largely functional still. But let's go back to the summary of what we found, the differences we found between these two species. If we remember, this enrichment of duplicated genes and repetitive elements actually match our expectations of what we might encounter in facultative symbionts. And this is a facultative symbiont. But then now that we know that symbiosis can impact genomes and how they change over time, how does this translate into diversification? For that, we can consider each of the changes separately. For instance, gene duplications can contribute to changes in gene families, as we observe or as we didn't observe. But some of these gene copies can acquire different functions, of course. Structural rearrangements contribute to the loss of conserved synteny among these genomes, and so do mobile elements, right? That further disrupt the synteny, but they can also impact gene structure, and they can also contribute to acceleration of mutation rate. And more interestingly, all of these changes are not restricted to certain regions of the genome, like for sexual recombination, where changes only happen in homologous regions, right? Or regions of high similarity. They can really happen at any scale and at different places of the genome. And if we think a bit more about this, some of these changes can have direct impacts on the fitness of these algae. And that's what I've done along this line for now. But of course, I would like to keep working on this topic in the future. And these are just some of the plans that I have. Of course, I would like to generate additional high quality genome data from other symbionts and free living taxa in the family. I would also consider the effect of divergence time in these genomic changes. And I think what the convention in the community, or what we think, is that the last common ancestor to all Symbiodiniaceae is symbiotic. But really, this is not conclusive because, as we mentioned, there is pre-living taxa across the tree that make that, of course, not final. And that's something that I would like to look into. I would also like to test formally the association between symbiotic lifestyle and diversification rates. And also, this is another experiment that I would like to do to try to track genomic changes of Symbiotic both in culture and in hospite over multiple generations. Something that I would like to do in the future, but this is in a longer term, is like to branch out to some other symbiotic systems, right? Some other microbial eukaryotes, which are plenty out there. And I actually have some ongoing projects along this topic still. So here at Penn State, starting on working at establishing this protocol for doing whole molecular weight DNA extraction from the symbionts directly from host tissues. And the idea is just to enrich the symbionts somehow, probably with a centrifugation gradient. And then also lysis can be tricky. So we're gonna try different things like sonication and just liquid nitrogen grinding. And then just run like standard, like traditional methods to get high quality or high weight molecular DNA. And this is something that I'm working on with this amazing undergrad student at the La Jeunesse lab. Also, to account for divergence times, I've spotted 2 genera that contain symbiotic and free living species. And more interestingly, these 2 genera have diversified over very different timescales. So that would be an interesting comparison. And these genomes are actually currently being sequenced by efforts of the Aquatic Symbiosis Hub from the Moore Foundation, where also the Sanger Institute is supporting. To test for symbiotic lifestyle and diversification, I plan to do use this approach based on ultra-conserved elements of the genome that pretty much give us also genome scale information, but at lower cost. And we can use this to reconstruct the evolutionary history of different traits. In this case, I would be interested in the symbiotic lifestyle to see which one is most ancestral. And we can also use this information to estimate diversification rate across the lineages. And then we can run some types of correlation tests between these 2 different traits. And this is something that I've been discussing with Andrea Quattrini, and we're working on that. But wait, there is more, because since I arrived here at Penn State, there are so many amazing things happening that I'm really excited about, and I want to share with you a few of the things that we're doing here. So one of the things that we're working on right now is on generating the genome. Well, we already generated the genome sequence of this symbiont, and this is a very interesting symbiont because it's a symbiont of giant clams, right? And these giant clams are really, really big, as you can see here. And they also can have beautiful patterns, beautiful colors in their mantles. But this system is very interesting because it's a case of extracellular symbiosis. So we don't really know how the genomes can look that within the context that we just talked about. They're also thermotolerant species, and they are basal lineage to an adaptive radiation in the Indo-Pacific. Also, microsatellite data has suggested that these species underwent whole genome duplication. So there are really many angles from which we can study this one genome. And this is something that I'm doing in collaboration with this amazing team of investigators, including Todd here. And actually, the person in the picture is Todd. So anyways, what we found so far is that, well, we look into specific genes. Genes are only occurring in this one species, but sadly, most of these genes have no functional annotation. And this is a common issue when we study Dinoflagellate genomes. We don't really know what they're doing. And yeah, so that's a bit sad. I was also running some selection tests, and I couldn't find any gene under positive selection, but there are some genes that are under, probably under some relaxation of selective pressure. And here we have something that is significant, is methylation, which is interesting and might be worth to look further into. Then we also look at functions, genes and gene functions associated with an intracellular lifestyle that we wouldn't expect in the extracellular symbiont, but we really didn't find any interesting pattern, and same for stress response. So these might be regulated at a different level that is not the genomic level, like epigenomics or post-transcriptional regulation. In terms of genome duplication, we didn't really find any evidence of whole genome duplication either, but instead we find a lot of repeats in the genome. So probably this is most likely what we saw before with microsatellite data is probably caused by extensive segmental duplication. And this is something that I am exploring further, not only for this genome, but for other Symbiodiniaceae lineages with this brilliant grad student, also from the biology department, from the other groups. Another project that we're working on at the Lajeunesse lab is looking at transcriptomics of host generalists. And here we're looking at 2 different species that associate with the same 4 coral species at the same location. And we want to see patterns of global gene expression, right? And how these symbiosis work. And this is a project that is being led by this grad student that is actually sitting right here, Caleb. With the Medina lab, we're also working on some other projects, and this is one of them. And this is actually led by Eric Crandall. And here we're looking at hybridization of Caribbean corals. And in the Caribbean, there is this species complex that is very interesting, right? Where it appears that there has been a lot of hybridization. And we want to assess what's the extent of that hybridization broadly in the Caribbean and the gene flow from a population genomics perspective. And this is again led by Eric Crandall. And also we have this grad student that's starting up looking into this. There are also some other cool things that we're doing. We're trying to mix ancient DNA and metagenomic approaches to try to trace back history of how, for instance, human activities have impact for our reef health. And for that, we have this genome core that we're trying out, that we sequence, and we're looking at fluctuations, changes in the different fractions of the holobiont, so the coral host, different algal symbionts, and hopefully we're gonna do that for the microbiota too, right? And this is something that is being in collaboration with the ancient DNA labs here at Penn State. We're also looking at some microbiome analysis. Particularly, we're interested in the microbiome of the upside-down jellyfish and the symbionts, which are also Symbiodiniaceae. So it's very interesting because to transition between some life stages to metamorphose, these jellyfish really need to be symbiotic to do that. So we're going to see how, and we can see the different types of symbionts have an effect on that metamorphosis. So we want to see also what is the added effect of microbiome and maybe play with some synthetic communities. And that's going to be led by Vivian, also a grad student, a new grad student that is sitting right here in the Medina lab. Also, this is a project that I'm also looking forward to do because we're going to get this started. I've never worked with single cell sequencing data. And this is something that we're planning to do again on the upside down jellyfish. The idea is to generate a cell atlas and also see changes in gene expression in different tissues, in different organs under different stressors. And this is something that is going to be led by the senior grad student, that's Victoria. She's sitting in the back, I think, again, in the Medina lab. And with that, I would like to finish and thank all the amazing people that has helped me throughout my academic career and here at Penn State, even though I've been here for a short time. And thank you for your attention, and I'm happy to take any questions. Yeah, thank you.